All right. Hello, everyone. We are the AR protein visualization team. This is our team. This is our team. My name is Veer.
I'm Alex.
I'm Sam.
And for those of you that don't know what AR stands for, AR stands for augmented reality. And essentially what that is the user's 3D world, essentially their real space, has computer generated images sort of superimposed into their real world. It's kind of like the halfway point between a virtual reality and their actual physical world. And then the reason why we chose augmented reality to visualize our proteins is because viewing 3D data on a 2D computer screen kind of misses out on important details. And then visualizing a 3D model in an actual 3D space just provides a more immersive and kind of interactive experience. And then that kind of leads to our project, which is our goal was to essentially create an AR environment to visualize and analyze molecular proteins. And this way users can kind of use it for like research purposes or just simple educational purposes. And our data source for this protein project was the Rutgers Protein Data Bank. And then the tech we're using to actually visualize it is the Unity game engine with C# scripting. And then to actually deploy ourselves into the AR environment, we're using the Microsoft HoloLens 2. So here's kind of a program breakdown of how our code works or how our program works from like start to finish. On the left, you can see Alex actually spawning in a protein. But essentially, when a user loads into the program, they'll be present with the text box for which they can enter like a 4-character code for the name of the protein. And what that does is it'll go to the database and download the CIF or PDB files, which we'll go into more detail later. But essentially, that just contains a text file with important information about the visualization of the protein. And then with that information, we create a CSV, which kind of filters out the useless information and extracts only the important information, which then our game engine will do the complicated stuff like creating the 3D model and coloring the atoms based on like the element and also combining the meshes so the model loads under one model only.
So getting into more detail here, first, it has to find a protein databank file or a crystallographic information file. Now, the protein databank file is the industry standard for the protein data, except sometimes only about 80% of proteins have a PDB file. So proteins with incomplete data will have a CIF file instead. Now, whichever file is found will then get cut down to a CSV file, which is commonly used in Excel, so the program can read them faster. So then from there, the program will read through the CSV file and generate the different structures like the atoms, atom bonds, and you can see the blue lines are the hydrogen bonds. Also beta sheets, which just represent peptide sequences, and also helices and RNA backbones, which we discovered by detecting phosphate sequences in the atoms. Now, we ran into two major roadblocks. First, Unity doesn't have any built-in curves, so we had to initially, the helices looked very jagged and disconnected from each other. So by using geometric curves, we were able to make them look like flow better, and also we were able to find the residue sequences to create those extra ribbons you can see. Secondly, some proteins have over 100,000 atoms in them, and generating that many individual objects led to major performance issues, and some proteins took up to 10 minutes to load in. Therefore, we loaded them in chunks, which greatly improved performance. So as you can see on the left, that is what a protein would look like using professional software like PyMol, and on the right, this is what our Unity recreation looked like.
So one of the most important features that we've implemented is the use of voice commands through the HoloLens. So we wanted to give you guys a quick demo on that. One thing you can do with the voice commands is be able to isolate different parts of the protein, and that's the first video.
Helices. Show atoms.
Here, Alex is just repeating different voice commands, so showing atoms, showing the connections, showing different covalent bonds, etc.
Hide protein.
And we also implemented the ability to isolate different types of atoms based on the elements they represent.
Show nitrogen.
So this is nitrogen,
Show oxygen.
oxygen,
Show water molecules.
and the water molecules.
Show protein.
So in the future, one of the things we want to do is implement greater detail into our structures. Currently, as we mentioned, we're not pulling all the information that's available within the PDB or CIF files. We extracted what we thought was most important, but it would provide a much smoother structure if we pulled more information from those files. And as Alex mentioned, we want to continue optimizing the rendering time as well as the model generation for the proteins. We were able to mitigate this issue a little bit with the batch implementation that Alex mentioned, where we implemented proteins in chunks, but this could definitely be improved. Another thing is being able to deploy these visualizations to some sort of mobile application, because the HoloLens isn't exactly the most accessible device, and to be able to put this on, say, a phone would be really helpful to be used in different types of settings, not only a lab setting. We also want to get feedback from professionals in the industry, see what they could use this for, and based on their feedback, be able to improve our technology. So what could this project be used for? One of the most important things is interactive learning experiences in a classroom setting. Students would be able to interact with different protein structures, see the bonds, be able to put different proteins together and see how they interact. Another thing is drug discovery as well as medical training. We were lucky to be able to speak to Professor Ashley Go from the chemistry department at Rutgers. She mainly works in molecular visualizations. That's what her research is based on. She told us that using technology like this would be really important to discover different bond sites and see how the drugs could be manipulated. Lastly, just public health awareness, being able to put this on, say, a mobile app like we mentioned, and being able to put this in hospitals or museum settings would really show the general public about different current health issues. For example, we have the COVID protein right here, and we all know that's still pretty relevant. We really want to thank Yvonne Seskar, Jennifer Shane, and our professors Wade Trappy and Ashley Go for supporting us throughout this entire process. And thank you guys for listening. Any questions?
Yeah, it's all in real time. So whatever like kind of like protein the user wants to visualize, they can do it while they're in the program, and they can also just load multiple ones if they wanted to. Doing it on both. We have our GPU kind of instantiate the objects, and then our CPU is what loads it. Yeah, later on when all the presentations are done, we're going to be doing our demos over there, and then you guys can come around and kind of see how it works and spawn your own proteins in if you want.
Thank you.